Good afternoon to everybody. We are happy to have here a very pioneer scientist, Professor Benjamin Wolozin, Ben, as we all like to call him. He's among of the, I would say, the more brilliant people I've met in my life because he can make ideas and make vision for students, for research fields to open, not only in Alzheimer's disease field, which we like more, I like more, but also in Parkinson and other diseases. He has been for many years a professor and created a lot of PhD students. He has a huge CV. I don't want to go through that, with a lot of successful funding and a lot of postdocs that were in the field successfully continue as group leaders. So all I had to say, we're very, very happy to have Ben in the HIAAD Bridge seminars with us. Ben, I don't want to waste the time. The floor is yours. I know you're going to surprise us with new stories, and we're very happy and looking forward to listen to your stories and great data.
Great. Okay. Well, John, I want to thank you for the invite and the opportunity to present. It's super exciting. Let me just hide video panel. There we go. Oops, I didn't want to do that. Show video panel. Sorry, hide controls. Okay, there we go. So you wanted something new. So I thought I have been working for about 5-6 years now on this project through a couple papers, but it's finally come out with we finally got to a point where I think it's really a tool that I think is going to be valuable for widely across the scientific sphere. And so I wanted to present this. So the title of this talk is Unbiased Characterization of the Evolution of Protein Complexes with Aging and Tauopathy. First, as always, I need to present my conflict of interest. I'm Chief Scientific Officer, co-founder of Aquina Pharmaceuticals, and we're developing small molecules to treat Alzheimer's disease and ALS, and we're actually putting in an application for one of our lead programs to the Michael J. Fox Foundation also, so hopefully Parkinson's disease too. So, through the years, the field of Alzheimer's and in general has used increasingly sophisticated proteomics to analyze the brain and the disease. So you have early kind of mass spectrometry, just measuring things, and then you have 2D page gels and with mass spectrometry, quadrupole, triple pole mass spectrometry. Then we went and we started, developing TMT so we could measure multiple samples, and we developed SILAC, and then there's other methods. So phosphoproteomics. So we have been able to quantify levels of proteins with increasing detail. Now, we're doing single cell proteomics. And then in addition, there are these kind of proximity profiling methods that have been evolved that allow you to say, well, I'm interested in this protein. I wonder what's directly binding to it. And that has been incredibly powerful. But these proteomics suffer, or not suffer, but they're limited in an essential way. When you think about biology and about proteins, knowing the level and the modification of proteins, that's important. But the reason there's been so much interest in things like proximity profiling is because fundamentally, we want to know what's touching what things are interacting. But as powerful as proximity profile labeling is, it's actually has a limitation because you shine a light on a protein and you look around that protein, but there isn't any way of, on a global scale, saying, I want to look at a lot of complexes, even for proteins I wasn't thinking of, and see what's happening with them. So my colleague, Andrew Mealy, developed this method in 2019 called EPIC. It's a toolkit for elution profile-based inference of protein complexes. That's a mouthful. So what does that mean? What that means is that what his lab did, and they did this on nematodes and yeast, you take proteins and you run them through through ion exchange columns. So you get these kind of co-fractionation profiles where, there's lots of kind of complexes. You all have kind of remember doing ion exchange. There's lots of complexes that come out. And you run those through mass spectrometry. And so for each complex, you know exactly, the range of proteins in the complexes. Then what Mealy and colleagues did was they took these complexes and they ran them through a machine learning algorithm that pulled in complexes from all the databases out on the web to identify kind of a global omic kind of signature of what are complexes out there. And then they use that to apply to these coelution profiles, so that you could say, oh, if these two proteins are here, it's probably this complex. So I'm simplifying too much, but based on the proteins that are here, these are the complexes that are appearing. And they took that and they validated it strongly using nematodes to show that, in fact, what they got out of this was some inferred complexes that actually predicted functional activity in the worm. Then in 2020, I got together with Andrew, and we were like, let's go and try this on the brain. Let's see what complexes come out of the brain. And so we took mouse brain and we ran it through these ion exchange columns, and then we went and Andrew his team did mass spectrometry on all of these complexes, ran it through the EPIC profile, and out of that we got about over 1,000 complexes. Note, these are soluble complexes. These are things that you can run through an ion exchange column and get information on. So there's definitely stuff we're missing, but this is a good start for being able to look at complexes. And in fact, those who know me know that I really like RNA binding proteins, and they show up really nicely in this system. So here's the workflow. You take, mice so what we did, sorry, 1 second, I think, yeah, sorry. I'll come back to that slide. Apologies. Well, let me just go here and show this. So what I'm going to show you in a little bit is application of this workflow to MAPT mice. But for now, what we did was we took these brains, we did co-fractionation, we did the mass spec, we did the inference to identify kind of complexes, and then we looked at what the complexes were. And so in this initial paper, we identified over 1,000 complexes, and here you can see example of like what one of these complexes looked like. We then went, and when you look at these complexes, what you can see is if you take fractions from different brain areas, the complexes vary based on brain area. Now, I will admit to you that when this work was first being done, I was highly skeptical. And I have always been highly skeptical of this, but my attitude towards this changed when I was at a functional genomics conference recently, and people started talking about these genes that they had found, and they didn't know what they were doing in disease, and I would just be like flipping through my files, and I'll show you them later, looking at them, seeing what they were. And for example, there was this one protein, LACT-B, number 2 hit in omics kind of informatics analysis of AD, and the people at Mount Sinai were saying, well, we don't really know what this does, but it seems to be important for some reason. And I looked and like, there it is. You can see it's interacting with mitochondrial proteins. And then the next slide said, the only thing we know is it's associated with this one mitochondrial protein, but we don't know why. And, like that type of thing made me think this actually is highly predictive. So here this shows when you run this inference of complexes from different brain areas, you get different patterns, which suggests that there is, in fact, some type of specificity to it. And you can also see that you get patterns that differ based on predicted cell type. Importantly, in this first work, we took some of these complexes and we looked at a TDP43 complex, and we looked to see and say, is this does this predict something that we would expect? And we saw that this complex had TDP43 and it had ALOV, it had DDX5, HNRMP, H1. Here's TDP43. It also has TIA in it, which I like, but and we were wondering, like, is this actually, for instance, do these actually functionally interact? So we did immunoprecipitations, and in fact, if we immunoprecipitated TDP43 or HNRMP H1, we found that we could find it associated with each of these different proteins, suggesting that, in fact, when you look into it, this complex is indeed something that is at least reproducible. Sorry, I'm sorry that this is the nature of having a new talk. That there's one slide. Yeah, there we go. Yeah. So apologies for, this is the first time I've given this talk. This is a paper. The stuff I'm talking about is we're about to submit imminently. So I'm going to mess up a little bit with it. But basically, we looked at this complex, TDP43, DDX5, and HRMP-H1, and we asked, are they functionally related? And so it's known that TDP43 regulates SORT1, and there's a cassette that you can do to look at the regulation. And so we took this SORT1 and we looked at it either, you either get these 2 exons or you get exon 17B, and TDP43 functions to prevent splicing of exon 17B. So you can see here that if you take cells, you express this cassette, and you knock down TDP43 that you get increased splicing of 17B, increased inclusion. And if you knock down TDP43 and DDX5, you get even more, even though DDX5 by itself, does a little, but not much, not statistically significantly different. And HNRMP H1, again, doesn't do anything on its own. But if you do these together, then you find that actually HNRMP H1 blocks this complex from acting. So when you get rid of it, now you have HRMPH1 that prevents 17B inclusion, so that when you get rid of HRMPH1, you're removing that splicing. So that's showing that these complexes are indeed functionally active. So now let's go and take a look at the brain. So those of you who know me know that I like to study tau, and I come off of, what is we have to come off of what's the amyloid cascade hypothesis. And so the idea is you get A beta stimulates reactive microglia. Together with those, they cause damage to the neuron, which induces tau pathology. So, and we've always hypothesized that tau interacts with stress granules to promote a translational stress response. So the idea is that this whole stress response happening. The question is, that's a tau-centric view, and there's no question that tau is driving this. But the question is, what else is going on? Can we shine a light across and look at other complexes? So we took a PS19, P301S PS19 brain that expresses P301S MAPT and gets, lots of pathology that increases with age. And we went and we did proteomics, and we also did phosphoproteomics on this. Okay, and the phosphoproteomics reproduced what Judith Steen has shown, and what everyone knows about this, with the classic markers of phosphorylation being identified. Okay, that's good. So next, we did the proteomics, and we saw, again, what you would expect. You kind of see early change, see many proteins that have previously been associated with changes in the brain. And when you look at what these correspond to, you can see that you get kind of synaptic changes in the early and middle stages, and cytoskeletal changes. And then later in the disease, you start getting the immune response happening. So this is all to say the system is behaving. We also looked at the phosphoproteome. So a number of people are doing that, and we've done that too. And what one sees with the phosphoproteome is kind of, again, things that you would expect. You see large cytoskeletal changes with disease, and you see large synaptic changes with disease, including some things, that have long been interested in, bassoon, synapsin, one of the things that I'll mention later that stands out as being highly phosphorylated is DPYSL2, which is something people haven't paid attention to, but it's a collapsin that controls axonal resilience. So, we took this proteomic system and we applied the EPIC algorithm to it. So just to remind you, that means that we took the brain, and we, in this case, for this study, we took one one kind of brain area with pathology, frontal cortex. We ground it up. We took the soluble proteins. We ran them through an ion exchange column, and at this point in time, we have improved the system. Back in 2020, we ran it through first a cation and then an anion exchange column, and then did the mass spec and machine learning. Since then, we've determined that actually if we used a mixed ion exchange column and just captured things that were charged and then ran those through mass spec and epic, it actually produced an equivalent readout, which speaks to the power of the machine learning and being able to predict the complexes. From there, we went and we characterized the complexes that occurred at early, middle, and late stages of disease. And from that, we got over 4,000 complexes that show an evolution with disease. But a number of those complexes appeared to share some similarity, and I'll show you what I mean in a little bit. And so what we did was we ran it through an algorithm that looked for shared similarity, and you can set the bar at where the threshold wherever you want. But for the field, a classic threshold that's set is if complexes are 80% or more similar, they're determined to be probably a shared complex. So we merged some of those complexes to kind of reduce the number and to allow us to look at the interaction, the kind of evolution of complexes better. And so once we did that, we got 3,000 complexes, and that is set at the threshold of 80% overlap. And this is what it looks like. Imagine, I'm just giving you a snapshot. This is an Excel spreadsheet that I have that lists all the merged complexes. And so imagine this goes, from CID complex 38 up to complex, I think it's like 3,118. Okay, and then you can look, and for every one of these lines, we now have heat maps that show what's in the complex, how that complex changes with disease for each of the members of the complex, what's the level of protein for each member of the complex, and what's the phosphorylation state for the protein for each of those complexes. And so to give you a sense on what you kind of look through, I'm not going to talk in any further about this, but it's one we had looked at, and it's kind of fun to look at, which is bin 1. There's several bin 1 complexes, but here you can see, here's one, CID 40, and you can see it has these proteins. And if you look down, here's another bin 1 complex, and it has some of these shared proteins, but you can also see then it has proteins that differ, all right? And so you can look at these complexes, and this is good for hypothesis generation. You can say, oh, I wonder if bin 1 is interacting with this protein, and how, what is it doing to interact with the protein? And how does that change with disease? What's the function? And so I'll show you in a moment the type of data that one gets out of this. But you can see that imagine being able to look up your favorite protein and see, what might it be interacting with? And then you can generate hypothesis-based experiments based on that. And I will say, in my experience, this has a high degree of accuracy, both because you look at it and you find proteins that when you look in the literature, they're in there, but then you also find new proteins that when you look, you find they actually do interact, or later on someone shows they interacted. So we looked at 3 types of complexes in detail to begin to kind of see the degree of validity that these complexes have. Could we validate them? So here's 3 complexes. One contains a MAPT binding protein, another contains actin binding proteins, and another contains reticulin 3. These are all things that reticulin 3 is a risk factor for AD, MAPT we know about, and the actin binding proteins are interesting because they show up a lot in this database. So if you look here, what you can see is that how with disease these complexes change. And here, this is kind of showing you the level of each protein in the complex. And so kind of here's MAPT, and you can see some proteins like the pyridoxines that are redox proteins increase dramatically with disease. Other proteins like this LDHA and GD, sorry, it's hard for me to see, GOT1 decrease with disease. I'll talk about these kind of a little more in a little bit. And here we go. We see dynamin, which is known to be involved in itself is a risk factor for disease. We see profilin 2, which although it doesn't cause ALS, profilin 1 does. We see the synaptic proteins, synapsin 1, etc. When we go to the actin binding proteins, now we see these actin binding proteins, ABI1, NCK-AP1, WASF is in here, CFIP, which binds FMR1 and tethers it to actin, is in there, and you can kind of see these proteins and how, for instance, WASF2 is increasing with disease, ABI1 increasing with disease, CFIP is kind of goes up and then goes down with disease. And then finally, reticulin 3, which is a kind of Golgi-related protein known for regulating vesicular transfer from the Golgi to the membrane, as is required for A beta synthesis. That's why it's a risk factor for AD, and regulating autophagy. And it interacts with these TRAP complex that also regulates Golgi vesicular trafficking. And you can see how the TRAPs change with disease here. And so a different way of looking at it, and actually more informative, becomes when we look at these heat maps. So if you go back, every one of these columns, corresponds to a different merged heat map. And we have a database that has all those heat maps that one can get. And as the paper goes out, which will probably happen this month, that database will become available. And so you can see here's a MAPT complex, and here's MAPT. I will say right off the bat, one immediate way to think about this complex, remember that we are getting the soluble protein complexes. That means that as proteins in disease aggregate, we lose them from our vision. Because what you would expect to see is an increase. So, sorry, let me explain this. Each line corresponds to a different age, 3, 6, 9 months. This is the complexes, column fractionation mass spectrometry with the EPIC inference. This is the protein levels. This is the phosphorylation levels. And when you merge them to get the significance, this shows how that changes. And each of these little white dots shows where you have significance. I will say something that we struggled with was how to do the normalization. we normalize these. These three are normalized on the same level, which means that really the color is driven by the merged complexes, which change the most. That is one can argue with, but when you do these on separate scales, it does not provide much more information. So we thought it's of greater value to understand the proportionate changes in each. I'm open to suggestions on that. And then the merged complex brings these three together with its own normalization. So this is phosphorylation, total protein, and complexes. So you can see here, for instance, that here's MAPT. So we think of MAPT as increasing in phosphorylation with disease. That's true, but as it becomes phosphorylated, it becomes aggregated, disappears from this analysis. So what we're left with are essentially the active complexes of MAPT. And you can see the extent to which MAPT, it's interacting immensely with this DPY-SL2, which is this collapsin controlling axonal integrity and resilience. And you can begin to see key factors that are changing with disease and perhaps regulating the complex. So ENO1 is an enolase. As I mentioned, the pyridoxine 1 and 2 increase greatly with disease. These two do. And the opposite happens for the proton pump ATP-V1A, ATP 6 V1A, and which decreases, and you can see other ones that decrease, like SNAP91. And at the same time, you can also see which things are being phosphorylated. So in this complex, we're not worrying, even though these are changing with disease, they're not really phosphorylated. Even though these are changing with disease, phosphorylation is not critical. But we can see that phosphorylation of synapsin-1, MAPT, and DPY-SL2 appears to be the things that are correlated with complex formation. Similarly, if we look at the actin binding proteins, we go and you see how these actin binding proteins AB1 and WASF increase with disease, whereas a kinase that actually regulates RNA binding proteins, SPK2, decreases with disease. And here is CFIP, which is, I guess because I like RNA binding proteins, I like CFIP because it binds FMR1, so I think that's interesting. But I don't have any other reason to like it. And you can see where the key levels of control here are. So CLIP1 is important for being phosphorylated. It changes with disease. You can see that ABI1 is phosphorylated, and that changes with disease. And then PTR1 also changes with disease. So you can look at this complex and now kind of predict where phosphorylation is going to be important. And finally, when you look at the reticulon complex, here's reticulon 3. You see it's increasing with disease, and you see that it's becoming associated with trap C4, whereas a different trap is reducing with disease. It provides you an insight into how to progress. So here are the key phosphorylation sites that are changing with disease, here, here, and here. So these are complexes where they fit with what we've known. There actually is one other paper published recently that shows the interaction of MAPT with DPY-SL. So this confirms that, but provides a lot more information. We also can begin to look at disease-related genes that are linked to disease, and say, well, what happens there? So, for example, this PTK2B is a risk factor for Alzheimer's disease, and these interactions are largely, if you look at the literature, this is a well-known complex that has been validated by multiple people. So we know this complex is something that exists in the literature and is regulated, and we can now see like where the phosphorylation occurs, which is on PTK2B itself, as well as CRK, which is a different kinase. But now we look at another complex. This has not been described in the literature, PTK2B, and we see that it's interacting with all these proteins that are linked to translation, to RNA translation, AMP1, and all these tRNA ligases, HARs and CARs, and then over here, RPMS3, also linked to translation at the synaptic membrane. And so you can see where the phosphorylation sites are here. So I come back to this, so now this is identifying potentially a new complex. And the question in your mind is, how much do we believe this? Should we believe this? And I'll show you in a moment some functional data that really speaks to the kind of relevance of these complexes, and suggests that when you see a complex in here, it really is worth paying attention to. Here's PCOM, which is, again, of great interest to people, and you can see how it evolves. I don't have anything brilliant to say about the PCOM complex here, but when we go to TDP43, you can see that, as expected, it's interacting with a number of RNA binding proteins. And this is interesting because, remember, this is a tauopathy mouse, and there isn't a lot of TDP43 pathology in a tauopathy mouse. But the question is, how is TDP43 responding to the disease? Is it responding to the disease? Does it change with disease even though it's not aggregating? The answer is a clear yes. It shows, so here's TDP43, and it's showing striking changes with disease, and more importantly, the proteins with which it's interacting, other RNA binding proteins, RALI, HNRNA, RNPH1, and FUS. All of these are changing with disease. And we can also look at particular places where phosphorylation becomes important. And so you can see phosphorylation of the EIF2 complex, makes sense. You can see phosphorylation of RALI, well studied, something perhaps worth looking at. And so this becomes something of strong interest. So how do we validate these? Well, the first thing we did was we said, all right, if we have these complexes, if we take a protein in these complexes and pull them down, do we see evidence of the interaction? And so here we first did the obvious, pulled down MAPT, and sure enough, there is DPYSL2 showing up in there. There is ATP6V1A, which was in the complex, showing up in there. Not surprisingly, there's synapsin showing up in there. Similarly, if we take DPYSL2 and IP that, what do we see? Well, we see MAPT, we see dynamin, we see these proteins that are showing up in these complexes. And you can start looking at the network of interactions of DPYSL across the disease. And so we know a lot about tau aggregating. Do we know a lot about how collapsin responds as axons collapse? No, we don't. But do it with the using this epic algorithm applied to these brains to generate these complexes. We now can begin to look at how these complexes change with disease, and we can see kind of, here's the DPYSLs. They're interacting with actin proteins, all the actin proteins. And then you see MAPT up here, and you can see how these integrate. We also took CFIP, not known to interact with MAPT, but in fact, you pull it down, there it is. MAPT shows up very strongly with it. Actin binding proteins show up strongly. WASF, another actin binding protein. C1q, synaptic pruning. John, you'll be interested in that since that's an RTIA paper, shows up with it.
Definitely, yeah.
So that suggests that it's possible to take the predicted complexes and look for direct interaction by immunoprecipitation. And of course, that depends on, I just want to make a caveat, which is that a benefit, in addition to identifying global networks of complexes, there's a plus and a minus of our approach. The plus is that you see many complexes. The minus is that we're not using detergent, so we're not pulling up the insoluble material, because that would break up the complexes. But because we're using very light very light dispersion, we can see proteins that are associated with each other that are not as tightly associated as you might need for immunoprecipitation in the presence of severe detergents. Now, one can say, what about the junk proteins that are sticky? And I think this is the value of artificial intelligence, because by training this against known data sets, the machine learning algorithm can identify the interactions that are high value in terms of function and likely, have been validated as real throughout the literature, and can kind of sort through things that are real and things that are kind of junk. I will say, in that spreadsheet that I have, the first 3 rows are really big, and I don't trust those first 3 rows. The rest of it really seems to follow, but the first 3 rows are I feel have too many, and I haven't put effort into trying to validate those. But if your complex is too big, I become suspicious. What disappoints me is when I look for granulin, it only shows up in those first 3 rows. So granulin, not informative. Now, we went and worked with Juan Botas at Baylor, and Morgan Stephens, a graduate student. And we went and said, okay, let's look at some functional validation of this work. And so they took 2 fly lines, one that had MAPT and one that had A-beta-42. And we asked about, what did these members of the complexes, what did they do to comparatively between the 2 lines? And the first thing I'll say is that if you look at, we looked at 11 different genes, and we looked at about, for each gene, we looked at between 4 and 6 different types of modifications, knockdowns, overexpression. And what you can see, what's striking, is that the effects on the MAPT line generally are much more robust than the effects on the A-beta line. And you can see it visually here, where we take 3 of these. If we look at WASF1, you might say, oh, 6L, it really, if you knock down an actin binding protein, of course you're going to have trouble. But in fact, if you do it in a, not shown here, if you do it in a line that is like just a control line to say, is there any effect generally of this? You don't see much of effect on climbing. This is this is a score where they're looking at climbing of Drosophila, and the MAPT and A beta reduce the ability to climb. And so what you have here is this is the healthy control. This is kind of tau, and this is the tau plus with the kind of experimental perturbation. And so what you can see here is, of course, if you knock down WASF1, the healthy animals, they can climb fine. If you express tau or A beta, you can see that their ability to climb decreases. But if you knock down WASF1, that actin binding protein, you can see it exacerbates the MAPT effect, meaning that it's kind of part of this normal complex necessary for maintaining activity. And if you knock it down from A beta, absolutely no effect. Same thing for dynamin. We know dynamin causes disease, but you can knock it, you can have a HET for dynamin and knock it, reduce it, and the animals are still functional. Add in MAPT, less functional. Add in MAPT and knock down dynamin, boom, they can hardly function. Whereas if you knock down dynamin in an A beta model, it doesn't really impact on it anymore. And similarly for DPYSL2, this is overexpression, okay? It's hard to do DPYSL2 knockdown because it actually causes collapse of axons. So to look at overexpression, but what you can see is when you've overexpressed DPYSL2, you are exacerbating the deficit caused by the MAPT. And whereas when you do it with A beta, you have no effect. So that speaks to the specificity of the complex towards MAPT and speaks to validate this massive massive database that we've generated to help us understand at least brain function within cells. So what I've shown you is I'm presenting this new approach, which we call CFMS, and it's built on an algorithm called EPIC that allows visualizing on a global scale how complexes change with disease. So you can look at this. You could essentially look at this, for any complex you want. Do the CFMS and the EPIC algorithm and see how the complexes change with disease, and that becomes hypothesis generating with a strong chance for that a fair number of those complexes are readily able to be validated. And you can also see how these complexes will change with disease as the disease evolves. And if you combine that with phosphoproteomics, you can see exactly what proteins in that complex have phosphorylation changes that correlate with disease, which then you could, query by doing site-directed immunogenesis and making, phosphomimics of, increasing or the null phosphorylation to test this. So, in conclusion, kudos to my colleague, Andrew Emili, who made this EPIC algorithm that combines ion exchange chromatography and mass spectrometry with machine learning to create a global characterization of soluble complexes. What's striking is that because the machine learning is so powerful, the complexes generated by EPIC have been strongly validated in nematodes. We have validated it in mice using multiple different orthogonal approaches. We applied EPIC to MAPT and identified over 3,000 complexes that dynamically change with disease. When we analyze these complexes, we take several complexes and look at them in detail. We're able to validate them, the members by IPMS, and we're able to functionally validate them in Drosophila models of AD. What's striking in the functional validation is that there's a striking selectivity for impacting on MAPT flies as opposed to A-beta flies. We integrate it with phosphoproteomics, which gives us sites for regulation. And then we drilled down and looked at a small number of complexes to really understand, to use it, to tell us something about, the brain. And so we identified this complex between MAPT and DPYSL2 that controls axonal resilience, is stabilization. That's what we think it does. We looked at PTK2B. We saw a known complex, but there's a bunch of complexes there, but we also saw complexes that included AIMP2, tRNA ligases, and I didn't mention this, but it's really it's something that's close to my heart these days, YTHDF, it's actually, I should say 3, which is a M6A binding protein. And so this suggests a role in translational regulation. And then finally, throughout this, we see all these actin proteins that seem to play a role in kind of maybe holding together and somehow are involved in many of these complexes. And we also see which proteins change with disease. So I want to give kudos to the people who did this. So Sophie, first of all, Andrew Amelie and his team really drove the work here in kind of terms of designing EPIC and doing the CFMS. We came in as this was kind of my idea to look at MAPT and doing the validation. Juan Botas at Baylor. Luckily, I'm not speaking at Baylor because I realized I left him off this list. Again, first time I've given this talk, so there's some mistakes. In my lab, Sophie Vanderspeck, Ali Rondon Ortiz, who actually just graduated and is at the Broad Institute, and Alex Taylor were essential in making this happen. Thank you.
Thank you so much, Ben. Really, really, as I was expecting, something out of the blue, changing a bit the way we can use and proteomics and mass spec in order to understand further the single proteins. Because this is what you tell us today, correct? Mm-hmm. We should not go to see single proteins that they don't tell us, like levels up and down, but to whom they interact, if they're affected, phosphorylation, interactome, and that can be more close to functional than the string, let's say, based on this type of connection network of pathways we were using. That's much
exactly more. And you can see, like, we all go and use string on gene cards, but it doesn't tell you how it changes with disease.
Yes.
Here you know exactly how things are changing, the complex is changing with disease, and if you include phosphoproteomics, then you know which are potential sites for regulation too.
Exactly. That's I think it's really novel. I see Joanna smiling because she's thinking something and Nodas is there too. But that's, I think, the innovative way, correct? That, and then we open the discussion to people. It's that you have included also the phosphorylation state in the same complex and how this is affected. And then we can answer questions that we have there for years. Where when you dissolve and break stuffs, you don't measure anything that interact or close or where it is inside. You just make a bloody soup, which does not really informative sense up and down or not change, but not change in phosphorylated states, bind it with the other.
Yeah, exactly. Yeah.
Wow.
So, I mean, and I guess what I will just say, what's really impressed me has been when I've sat in seminars and listened to other people speak and and just kind of gone and done this. Even I have to admit, maybe a little illegally, but I was doing this with my company where we had this target for a really effective Alzheimer compound, and we're like, and the target's something that no one's really studied that, and they're like, but the CEO is like, you've got it, but what's the mechanism? And I like open it up and like, oh, here's the mechanism. This makes total sense. It's just like, it just is really, I'm sure that, some of the complexes or some components of the complexes are not real. That's, that comes with any omic thing. But I think there's a lot that can be, it's great for hypothesis generating.
Okay, that's great. So let's open it to discussion. So, any questions, guys? Because I have plenty, but Nora, Joanna, Makis, I see here, Kostas, Oleg Karmias is also here. He has the translation and clinical work together.
May I?
I see Rebecca Bakat says a lot of this. Yes, Costa. Open your camera so we can see you.
Thank you very much for this very interesting talk and very impressive volume of work. Can you hear me, first of all?
Yes. Yeah, I can.
Yeah, very good, very good.
I see a few things. I'm an old timer, by the way.
I wouldn't have guessed.
Yeah,
you
you
don't look a
wouldn't,
day
but
over 20.
I am. I am emeritus researcher at the Institute, and I have seen the kind of co-fractionations of proteins and in exchanges, ion exchanges, chromatographies, and so on, coupled with mass spec work in the past. Not the type of studies you are reporting to us now, but I have noticed that you are using the term very highly, the term this protein, for example, MAPT or PTK2B and so on, interacts with. And you put all these proteins that are in the complex. And the question I need to ask you is, do these proteins interact with or are parts of the complexes that you are identifying?
You mean, are they touching
In other
each
words,
other?
is this, and that's one thing. So, for example, if one was to knock out the specific protein that is the basis of the interactions, what would happen to the complex? Would it fall apart, or nothing really would happen in addition to the disappearance of the specific protein that is being knocked out? And the second question that comes to my mind is whether these interactions are causative or consequential in terms of disease status. I'm not sure whether what I'm saying is clear to you, but
Yeah, I understand what you're saying. Let me first ask the question. Let me first answer the thing that I first thought you were saying, which you didn't say, which was, I thought you were going to say, are these things actually touching each other if they're in the complex?
yeah.
And the answer, I think, is not necessarily. It's just saying they're in part of a complex that's coming down together and is loosely associated. So they don't have to be directly interacting for them to appear, but they have to be, I like RNA binding proteins. It works so well. Like, you can imagine if you have some RNA and you have a bunch of, proteins that are phase separated together, they will stick together in a complex, particularly, if they're kind of a little bit gelatinous. And so and we see
you can also demonstrate function for these proteins by knocking out, and then you see and
So-
exons being skipped, and this type of work.
I
It
know
has done in the
exactly.
past.
I know exactly what you mean. And so in the brain map, we actually, in our first publication, 2020, we actually took I think it was an ataxin knockout mouse and like looked at the effect on a TDP complex. Because in that TDP complex, I showed you there's ataxin too, and that showed up in brain map also. And we knocked it out. In fact, it did change the complex. But I suspect it's going to depend on the protein. Again, if you think like with RNA granules, there's core nucleating proteins, and then there's the peripheral things that are just kind of in there. So I don't think one can say definitely that that every protein in the complex is necessary for it. Where things stand out to me is if I see a protein that is coming, like showing up highly into a complex, or being removed from a complex, and something else is changing at the same time, I would look and predict, hypothesis generating, predict that change is causative. And that's testable. It might not be true, but that's a testable hypothesis.
Exactly, yes.
Exactly.
We did Drosophila because for two reasons. The first was that it turned out atrociously hard to, every time we were trying to get the DPYSL2 in neurons, every time we knocked it out, the neurons would just collapse. And we couldn't do that experiment. In Drosophila, we could, and I'm part of this functional genomics consortium. So Juan has a high throughput system on BOTAS, so he could do this. But what we have seen consistently is that if you take members of this complex and you knock them out, you see functional effects. The functional effects that you see, though, this is true of Drosophila, this is true of when we were looking at the TDP43 splicing studies, depends on the particular component. Some components promote the complex, other components inhibit the complex. So I don't think you can predict the effect of the protein just because it's changing. You can do other things that were predicted, but
Yeah.
Thank you.
The machine
Question?
learning,
Costa, allow me.
Oh yeah, I'm sorry.
See,
Okay.
no, great discussion. I just want to see if there are no other questions, and then we can continue. I just want to that way everybody has a chance to ask. But great discussion so far. Do we have other questions, guys, from other people? Otherwise, we'll continue. I have no.
And I have one.
Makis Koulakis, a Tau Drosophila guy. I'm just giving a little introduction. Kostas was the previous director of the institute where I am now. Happy to have with us in the same team. Maki.
Yeah, so thanks for the very, very, very nice talk, and widen our horizons with tau effects. The question that I have is, of course, the tau mouse that you use is the P301L. The question is, would you expect the same thing, say, with a Davis mouse, or a mouse that doesn't express a human protein, a human tau with a mutation,
yeah. So I think that this paper, that this approach that we did here, does have in the paper, you send out like that, you always write that limitation sections.
Yes.
And so one big limitation section is soluble complexes. A second big limitation is, I'm making a big deal about the value to look at other proteins, like, essentially the endogenous proteins, and look at what's happening with them. And yet, what mouse, in order to mimic disease, what mouse am I using? This P301S overexpression mouse, which is not, so that MAPT complex actually think, actually that particular DPSL actually does have a lot of validity. But you could imagine seeing complexes that are because of MAPT overexpression. I think in the case of MAPT, because we are looking at because we essentially lose the pathological MAPT complexes from our study,
Yes.
we're actually looking at the endogenous functions of MAPT. So John, this is really up your bailiwick for the things you really want to know. And so that DPYSL2 complex, that's not one that increases to cause disease. It actually, as that goes away, as that complex becomes dysfunctional, that's what correlates with axonal collapse. So I think, example in that complex, that if I look in a Davies mouse, where, or let's say I take Sunosato's knock-in mouse, where we're not expressing it at all. But the knock-in mouse also doesn't develop pathology until very late in disease, right? You're going to see that same complex there because that's an endogenous complex. You will, and it might actually be more robust later in age than in these mice. In a sense, the weakness of the study, looking at soluble complexes, presents a strength because we're actually looking at the response of endogenous non-disease, kind of the other stuff involved aside from the light that we always shine on MAPT aggregation.
But do you think you might get complexes that are specific to the mutant protein
To the what?
versus the new?
Yeah,
Would you
the
get
new
complexes that are
protein.
specific to the mutant protein?
So to the best of my knowledge, the mutations in tau that correlate with disease act largely by increasing the tendency of the protein to aggregate. It's different than like a point mutation that affects the binding to a specific protein or something, or enzymatic activity. And so because of that, and because the aggregates are lost from our study, I don't think those are I don't think that the type of mutation will impact greatly. So I will say, Nandas, I did look at synuclein. There's two synuclein complexes there. One which is a head scratcher and one which seems to make a lot of, has, some stuff that makes sense, some stuff that's hypothesis generating.
Okay. Thank you.
More questions for Ben? Joanna? I have a little friend in this seminar today.
Who's your little friend?
That's
Yes.
a Portuguese friend, Luisa's daughter, that is with me in this meeting.
That's cute.
Yes. Joanna, do you have any question?
I have many questions.
Just
But
go.
I think I just entered in the middle of the seminar because I was in a meeting. So I was going to ask the same as Kolakis, which is if you think that this is just dependent on, this is true in physiology, not really in the pathology itself. And I believe it is because we are talking about these soluble complexes. And what do you think about these, the role of these soluble complexes that are formed and especially relates with synaptic function?
So there's a lot of synaptic proteins in these things.
Yes.
And by and large, it seems like the presence of these, based on the Drosophila studies. So by and large, the Drosophila studies suggest that when you remove a lot of these proteins, you make things worse.
Yeah.
That says to me that these complexes are by and large promoting homeostasis.
Mm-hmm.
So they are the kind of the endogenous workings of the cell that are kind of trying to keep functional in the face of cell damage. And so I think most of these complexes, if you remove them, are going to be deleterious, is my prediction. Here, and the big example I had of that was DPYSL2, where you really don't want to remove DPYSL2. That's what I learned. But it's there with tau. So
Yeah. And how local are they? Do you think they are important in their localization in the cell?
yeah,
You mean intracellular localization?
Yes.
Yeah, I mean, kind of following up with with imaging is clearly worthwhile. There's if the US had a functional government, I would say that there's a lot of opportunity for generating new studies and everything. I mean, we write the grants, but then nothing happens. So
wait. We need to be patient in life. A stupid technical question that they text me here. They text me here a stupid technical question. How difficult is to do the first part?
The CFMS.
And the MS is okay, but I'm talking about what is the difficulty to do the first part. So you have the iron of the columns, and how much tissue do you need to do that? For example, what you saw was from the cortex, hippocampus, was from different brain areas, and as well as would you expect based on hormonal changes or sex differences to have different complexes and different signal. I would expect, but I'm asking you what data you have in your hands on this.
So it's true
technical,
right. The CFMS needs to use a fair bit of tissue, on the order of, let's say, 80, to 100 milligrams of tissue. So you can't, it's not at the single cell level, right?
Okay, I didn't expect that. I didn't expect that. But I'm asking, okay.
But the flip side is, I'm doing a lot of work with assembloids now, and I can envision I can't do it on one assembloid, but I can take a plate of assembloids and do CFMS on that.
Got it.
So it's feasible, but it requires moderate, it requires significant but feasible amount of tissue. So, and it can be done with cell culture. It can be, there's a lot of ways. It's just not a kind of 96 well plate type of, like just what's in a 96 well plate. You need millions of cells. So, and then the actual CFMS, you run it on now one HPLC column. So you have to have the setup, but most of the mass spec people do have the setup, with an HPLC attached to a mass spec, so you can do that. And then the EPIC algorithm is on GitHub, so that's also, quite feasible. So I think that there's a lot that can be done with it, I think that's why I'm presenting it, because I just find it immensely useful.
Yeah. From as a platform and way of thinking and approaching that that's why I asked how easy it is, because I think it will be very innovative in the way we see our data or analyze or perform and analyze our data related to omics, proteomics. Probably there will be applications on different stuff, but at the moment for the proteomics, I think it's a new way of seeing dynamics and changes and networks moving to one in the other direction.
Right.
Okay. If we don't have more questions, it's 6:20. We're supposed to go for 1 hour, but as you see, there was great. Costa, do you have one last question? No.
It was just a small comment because I've got the last point about machine learning, and I think it's a great idea to put machine learning in there. But in my view, again, because I have done a little bit of this, machine learning conclusions should be tested in biological terms through functional assays. And
that's
then
right.
you will be certain that the machine learning algorithm that one uses is really a valid one. Or
I think
I totally agree. And that's why I actually sat on this for a long time before presenting it. It was actually when I was at a functional genomics meeting where I was like plugging things through that I was like really impressed by what you could see with it. So yes, it totally depends on this whole thing. These are inferred complexes that totally depend on the strength of machine learning.
anyway, thank you very much for
Thank you.
very interesting presentation.
Yeah, I would like to thank Ben for being with us today and all the audience and for the many people in YouTube. From me and my friend for today, we would like to thank you, Alicia, for being with
Alicia,
us
great to see you.
in another very interesting HIAAD Bridge seminar. Ben, thank you. It was an honor to have with you. And see you until next time, next month. Thanks.
Sounds great.
Goodbye to everybody.
And Joanna, we're as soon as we get that snRNA-seq, Christine said that's
Yes.
finally going to happen, and then that paper goes back out.
Great.
Okay.
Great. Thanks.
right.
Thanks. Have a nice afternoon to everybody.
Bye.
Bye-bye. Thank you. Bye.
Thank you.
Bye.